Welcome to the Explainer. Have you ever wondered how scientists can find the one specific gene that causes a disease, or how they breed a plant that can survive a drought? Well, the secret is in these tiny, invisible barcodes that are hidden all throughout our DNA. Today, we're going to unpack the story of these genetic markers, the incredible tools that let us read the code of life itself.
So here's our game plan for today. We're going to start with the absolute basics. What even is a genetic marker? Then, we're going to take a trip through three major eras of this technology, starting with what you could call a molecular scalpel, moving to the genetic photocopier revolution, and finally landing on the high-speed data firehose we have today. And to wrap it all up, we'll look at how scientists pick the right tool for the right job.
All right, so first things first, what are these genetic markers we're talking about? Well, you can think of them as the absolute foundation of modern genetics. They're the starting point, the essential tools scientists have to have before they can even think about analyzing a genome. It all comes down to solving this one massive problem. Think about it, a single genome can have billions of letters of code. So how in the world do you find and keep track of one specific gene, this tiny little needle in a ginormous haystack that you can't even see? The answer is with molecular markers. The best way to picture them is as unique, inheritable barcodes that are attached to specific spots on a chromosome. Now, the wild thing is, the markers themselves don't usually do anything, but because they're physically stuck right next to genes we care about, if you can find the barcode, you found the gene. It's like a beacon in the dark. So what exactly makes for a good marker? Well, they've got some really important things going for them. For one, they are everywhere, scattered all across the genome. They're also super stable. It doesn't matter how old the organism is or what environment it's in. And here's the really crucial part. They show a ton of variation between different individuals. That variation is the key. It's what lets us tell one person's genetic blueprint from another's. Okay, here's a really key distinction you gotta get. Codominant versus dominant markers. Codominant markers are like a high-resolution photo. They let you see both copies of a gene an individual has. So you can tell if they're homozygous, meaning two identical copies, or heterozygous, with two different ones. Dominant markers, on the other hand, are more like a silhouette. They just tell you that at least one copy of a gene is there, but they hide the rest of the detail. And that difference in information, it's a really big deal. Now let's jump in our time machine and go back to the very first generation of this technology. We're talking about a method called RFLP, and believe me, when it came out, it was a revolution. It gave scientists the first real way to dissect a genome with what was essentially a molecular scalpel. So how did RFLP, which stands for Restriction Fragment Length Polymorphism, by the way, actually work? Imagine you have two books that are almost identical. You use these molecular scissors that cut the paper every single time they see the word the. Now, because the books are slightly different, the scissors will cut in different spots, creating strips of paper with different lengths. By just measuring those strips, you can spot the differences. RFLP did that exact same thing, but with DNA. But this led to a pretty clear trade-off. On the one hand, RFLP was a fantastic tool. It was codominant and it was super reliable. You could absolutely trust the results. But, and this is a big but, it was an absolute beast to work with. The whole process was incredibly slow, it cost a ton of money, and it needed huge amounts of really pure DNA. It just wasn't practical for big projects. So you can see the problem, right? Scientists had this great, reliable tool, but it was just so slow. And that bottleneck, that need for speed, is what pushed them to find a better way. It basically kicked off a whole new era in genetics. And the answer came in the form of a technology called PCR, the polymerase chain reaction. And honestly, this was a total game changer. You see, PCR works like a genetic photocopier. For the very first time, scientists could take one tiny microscopic piece of DNA and just amplify it, making millions of identical copies in only a few hours. Suddenly, being able to amplify DNA like that just opened the floodgates. It sparked this huge explosion of innovation. A whole bunch of new marker technologies started popping up. But we're going to focus on three of the key players from this PCR-based generation. First up, you had RAPD. It was incredibly fast and cheap, which was great, but the results were kind of all over the place. They were hard to reproduce, which is a huge no-no in science. Then came AFLP, a really clever mashup of the old RFLP method and the new PCR tech. It was reliable, but it was still really complex to do. And finally, you had SSRs. These were awesome, super reliable, and codominant. But there was a catch. You had to already know the DNA sequence of your organism to develop them, which was expensive. So even though all this PCR stuff was a huge leap forward, it still wasn't perfect. All these little limitations kept pushing science towards the ultimate goal, the Holy Grail, a marker system that was ridiculously fast, totally reliable, and could give you an absolutely insane amount of detail. And that brings us to the third and current generation of markers, the SNP. That stands for single nucleotide polymorphism. Instead of looking at the length of DNA pieces, SNPs zoom all the way in to the smallest possible difference, a change in just one single letter of the genetic code. It's like going from reading whole paragraphs to analyzing a single letter. You just can't get any more detailed than that. And here's what's just mind-boggling about SNPs: how many of them there are. They are by far the most common type of genetic variation out there. That means the genome is just jam-packed with millions and millions of these potential markers. It gives scientists this incredibly dense, super detailed map to work with. But the real superpower of SNPs is how perfect they are for automation. Using modern DNA sequencing and these little things called microarray chips, scientists can now look at thousands of individuals and millions of SNP markers all at the same time. It is the undisputed gold standard for any kind of large-scale genetics today, even though getting started with the technology can still be pretty expensive. So we've been on this incredible journey of innovation, right? From these slow, clunky methods to this super fast tech we have today. So how does a scientist in the real world actually choose the right tool for the job? What makes them pick one method over another? Well, it really boils down to a series of trade-offs. Two things are super important. First, reproducibility. Getting the same result every single time is just non-negotiable for good science. And second is information content. This goes right back to that codominant versus dominant idea we talked about. Codominant markers just give you more data, which makes them way more powerful tools. And believe me, picking the right tool matters a lot. We're not just talking about abstract science in a lab here. These markers are the engines that power modern biology. They let us build the genetic maps we need to find the causes of human diseases. They help us dramatically speed up the breeding of the crops that feed the entire world. And they are absolutely essential for conservation, letting us check the genetic health of endangered species so we can protect them from extinction. You know, this table right here tells the whole story perfectly. Just look at the columns. You start up top with RFLP, codominant, highly reproducible, great. But then look at that throughput. Low. Then you see Rapide came along and tried to fix the speed problem, but at what cost? Low reproducibility. Not good. As you go down the list, you see this constant improvement, this balancing act, until you hit SNPs at the very bottom. Just check it out. Codominant, high reproducibility, high polymorphism, and high throughput. It's got everything you could want. It is the clear winner of this technological evolution. I mean, the progress has been just breathtaking. In just a few decades, we've gone from painstakingly reading one genetic barcode at a time to scanning millions of them in the blink of an eye. This incredible leap in our ability to see into our own DNA leaves us with one final, really exciting question to think about. Now that we can read the book of life with this kind of fluency, what amazing discoveries are waiting for us on the very next page?